Hello friends, today I am going to deliver a lecture on history of human genetics, mosaicism and chimerism, consanguinity and its effects, epigenetic modifications and imprinting. Now let us first try to have a look at the main objectives of today's deliberation. Number first, to give a general and thorough idea about the genetics. Number second, to discuss in detail about the human genetics. Number third, we shall be discussing in detail about the human genetics program. Number four, discussing in detail mosaicism and chimerism. Number fifth, we shall be learning about consanguinity and its effects. And finally, we shall be discussing epigenetic modifications and imprinting. Now, we will start with the brief idea about genetics. Genetics is the study of genes, genetic variation and heredity in living organisms. Heredity is a biological process where a parent passes certain genes onto their children or offspring. Every child inherits genes from both of their biological parents, and these genes in turn express specific traits. Some of these traits may be physical, for example, hair and eye color and skin color. On the other hand, some genes may also carry the risk of certain diseases and disorders that may pass on from parents to their offspring. Genetics form one of the central pillars of biology and overlaps with many other areas such as agriculture, medicine and biotechnology. The father of genetics is Gregor Mendel, a late 19th century scientist. Mendel studied trait inheritance, patterns in the way traits are handed down from parents to offspring. He observed that organisms, that is pea plants, inherit traits by way of discrete units of inheritance. This term, still used today, is a somewhat ambiguous definition of what is referred to as a gene. Now, let us talk about human genetics. Some geneticists specialize in the hereditary processes of human genetics. Most of the emphasis is on understanding and treating genetic disease and genetically influenced ill health, areas collectively known as medical genetics. One broad area of activity is laboratory research dealing with the mechanisms of human gene function and malfunction and investigating pharmaceutical and other types of treatments. Since there is a high degree of evolutionary conservation between organisms, research on model organisms such as bacteria, fungi and fruit flies, which are easier to study, often provides important insights into human gene function. Many single gene diseases caused by mutant alleles of a single gene have been discovered. Two well-characterized single gene diseases include phenylketonuria and Tay-Sachs disease. Other diseases such as heart disease, schizophrenia and depression are thought to have more complex hereditary components that involve a number of different genes. These diseases are the focus of a great deal of research that is being carried out today. Another broad area of activity is clinical genetics, which centers on advising parents of the likelihood of their children being affected by genetic disease caused by mutant genes and abnormal chromosome structure and number. Such genetic counseling is based on examining individual and family medical records and on diagnostic procedures that can detect unexpressed abnormal forms of genes. Counseling is carried out by physicians with a particular interest in this area or by specially trained non-physicians. Human genetics is the study of inheritance as it occurs in human beings. Human genetics encompasses a variety of overlapping fields including classical genetics, cytogenetics, molecular genetics, biochemical genetics, genomics, population genetics, developmental genetics, clinical genetics and genetic counseling. Genes can be the common factor of the qualities of most human inherited traits. Study of human genetics can be useful as it can answer questions about human nature, understand the diseases and development of effective disease treatment, and understand genetics of human life. Autosomal traits are associated with a single gene on an autosome, that is non-sex chromosome. They are called dominant, because a single copy inherited from either parent is enough to cause this trait to appear. This often means that one of the parents must also have the same trait unless it has arisen due to an unlikely new mutation. Examples of autosomal dominant traits and disorders are Huntington's disease and achondroplasia. On the other hand, autosomal recessive traits are one pattern of inheritance for a trait, disease or disorder to be passed on through families. For a recessive trait or disease to be displayed, two copies of the trait or disorder needs to be presented. The trait or gene will be located on a non-sex chromosome. Because it takes two copies of a trait to display a trait, many people can unknowingly be carriers of a disease. From an evolutionary perspective, a recessive disease or trait can remain hidden for several generations before displaying the phenotype. Examples of autosomal recessive disorders are albinism, cystic fibrosis, etc. Now, coming to the history of human genetics. Theories and studies in human genetics have a long history. Observations on the inheritance of physical traits in humans can even be found in ancient Greek literature. In the 18th and 19th centuries, observations were published on the inheritance of numerous diseases, including empirical rules on modes of inheritance. The history of human genetics as a theory-based science began in 1865 when Mendel published his experiments on plant hybrids and Galton studies on hereditary talent and character. A very important step in the development of human genetics and its application to medicine came with Gerard's demonstration of a Mendelian mode of inheritance in alkaptonuria and other inborn errors of metabolism. Further milestones were Pauling's elucidation of sickle cell anemia as a molecular disease in 1949, the discovery of genetic enzyme defects as the causes of metabolic diseases in 1950s and 60s, the determination that there are 46 chromosomes in human in 1956, the development of prenatal diagnosis by amniocentesis in 1968-69 for the detection of chromosomal defects such as Down syndrome, and a large-scale introduction of molecular methods during the last 25 years. Now, let us talk about the role of heredity and environment. The role of heredity and environment as regards the characters of an individual has always been debatable. In his efforts to find out methods of improvement of the human race, Francis Galton, a cousin of Darwin, at first tried to distinguish between effect of environment and those of heredity. He had introduced his famous twin analysis, that is, nature versus nurture methods for the purpose. He had shown that quantitative characters which had normal distribution were also inherited. After making observation on the generation of the Drosophila melanogaster, that is, the fruit fly, which has been the most popular insect for genetic research, Thomas Hunt Morgan had noticed that one of the flies had white eyes when red was their normal color. Studying the heredity of this trait, he could identify the gene responsible for this character in the X chromosome of the fly. In 1933, Morgan was the first of the illustrious geneticists to receive the Nobel Prize. Karl Landsteiner had identified the human variation in the blood group and the multiple alleles of the blood group genes, that is A1, A2, B1 and O. He had also discovered the MN and RH blood group. Since the inheritance of human blood group genes was practically pure and unaffected by environment. This discovery had provided very important tools for research in human genetics in addition to their practical day-to-day use in medicine. Landsteiner was awarded Nobel Prize in 1930. But it was Bernstein, the statistician, who had provided the genetic control of blood group in 1924. Important contributions to studies in the population genetics were notably by Ronald Fisher, J.B.S. Haldane and Sewell Wright. Fisher's contributions were outstanding in statistical aspects of agriculture and biology, including genetics. Haldane had contributed methods of gene mapping, ideas of linkage and calculation of mutation rates. Wright was the earliest to study the consanguinity, and he had also worked on selection. In 1940, A. E. Mursky and A. W. Pallister separated the DNA from the chromosome and found that the DNA maintained the same composition in all human beings. Now, let us discuss the next part of our today's lecture, that is mosaicism. In genetics, mosaicism describes the presence of two or more populations of cells with different genotypes in one individual who has developed from a single fertilized egg. Mosaicism has been reported to be present in as high as 70% of cleavage stage embryos and 90% of blastocyst stage embryos derived from in vitro fertilization. Genetic mosaicism can result from many different mechanisms, including chromosome non-disjunction, anaphase lag, and endoreplication. Anaphase lagging is the most common way by which mosaicism arises in the pre-implantation embryo. Mosaicism can also result from a mutation in one cell during development, in which the mutation is passed on to only its daughter cells. Therefore, the mutation is only going to be present in a fraction of the adult cells. Genetic mosaics may often be confused with chimerism, in which two or more genotypes arise in one individual, similarly to mosaicism. However, the two genotypes arise from the fusion of more than one fertilized zygote in the early stages of embryonic development rather than from a mutation. Mosaicism can be diagnosed in different ways. Sometimes, different cells in the blood have different chromosome makeups. For these individuals, a blood test may be able to detect mosaicism. In other individuals, it is more difficult to diagnose mosaicism. For example, a blood test would not be able to diagnose mosaicism if the chromosome change is only located in the skin cells. In cases like this, additional testing may be needed to detect mosaicism. Usually, this would involve examination of other tissues of the body, such as the skin. A geneticist can help determine whether this type of additional testing is appropriate. Different types of mosaicism exist, such as gonadal mosaicism, that is restricted to the gametes, or tissue or somatic mosaicism. Now, let us discuss the next part of our today's lecture, that is chimerism. A genetic chimerism or chimera is a single organism composed of cells from different zygotes. This can result in male and female organs, two blood types, or variations in form. Animal chimeras are produced by the merger of multiple fertilized eggs. In plant chimeras, however, the distinct types of tissue may originate from the same zygote, and the difference is often due to mutation during ordinary cell division. Normally, genetic chimerism is not visible on casual inspection. However, it has been detected in the course of proving parentage. Another way that chimerism can occur in animals is by organ transplantation, giving one individual tissues that developed from two genomes. For example, a bone marrow transplant can change someone's blood type. An animal chimera is a single organism that is composed of two or more different populations of genetically distinct cells that originated from different zygotes involved in sexual reproduction. If the different cells have emerged from the same zygote, the organism is called a mosaic. Chimeras are formed from at least 4 parent cells, that is, 2 fertilized eggs or early embryos fused together. Each population of cells keeps its own character, and the resulting organism is a mixture of tissues. There are some reports of human chimerism. This condition is either inherited or it is acquired through the infusion of allogenic hematopoietic cells during transplantation or transfusion. In non-identical twins, chimerism occurs by means of blood vessel anastomosis. The likelihood of offspring being a chimera is increased if it is created via in vitro fertilization. Chimeras can often breed, but the fertility and type of offspring depends on which cell line gave rise to the ovaries or testes. Varying degrees of intersex differences may result if one set of cells is genetically female and other genetically male. Now, let us talk about consanguinity and its effects. Consanguinity is the property of being from the same kinship as another person. In that aspect, consanguinity is the quality of being descended from the same ancestor as another person. The laws of many jurisdictions set out degrees of consanguinity in relation to prohibited sexual relations and marriage parties. The degree of relative consanguinity can be illustrated with a consanguinity table in which each level of lineal consanguinity, that is generation or meiosis, appears as a row, and individuals with a collaterally consanguineous relationship share the same row. The knot system is a numerical notation that defines consanguinity. In genetic terms, consanguinity derives from the reduction in variation due to meiosis that occurs because of the smaller number of near ancestors. Since all humans share between 99.6% and 99.9% of their genome, consanguinity only affects a very small part of the sequence. If two siblings have a child, then that child only has two rather than the normal 4 grandparents. In these circumstances, the probability that the child inherits 2 copies of a harmful recessive gene rather than one which would not have immediate effects is much increased. Conventionally, genetic consanguinity is expressed as defined by Wright with the coefficient of relationship R, where R is defined as the fraction of homozygous due to consanguinity under discussion. Thus, a parent and child pair has a value of R is equal to 0.5, siblings have a value of R is equal to 0.5, a parent's siblings has R is equal to 0.25, and first cousins have R is equal to 0.125. These are now expressed in terms of percentage of shared DNA. Now, let us talk about epigenetic modifications. Epigenetics is the study of potentially heritable changes in gene expression that does not involve changes to the underlying DNA sequence, a change in phenotype without a change in genotype, which in turn affects how cells read the genes. Epigenetic change is a regular and natural occurrence, but can also be influenced by several factors including age, the environment, lifestyle, and disease state. Epigenetic modifications can manifest as commonly as the manner in which cells terminally differentiate to end up as skin cells, liver cells, brain cells, etc. Or epigenetic change can have more damaging effects that can result in diseases like cancer. At least 3 systems including DNA methylation, histone modification, and non-coding RNA associated gene silencing are currently considered to initiate and sustain epigenetic change. New and ongoing research is continuously uncovering the role of epigenetics in a variety of human disorders and fatal diseases. The term epigenetics, which was coined by Waddington in 1942, was derived from the Greek word epigenesis, which originally described the influence of genetic processes on development. Currently, DNA methylation is one of the most broadly studied and well-characterized epigenetic modifications, dating back to studies done by Griffith and Mahler in 1969, which suggested that DNA methylation may be important in long-term memory function. Other major modifications include chromatin remodeling, histone modifications, and non-coding RNA mechanisms. The renewed interest in epigenetics has led to new findings about the relationship between epigenetic changes and a host of disorders including various cancers, mental retardation associated disorders, immune disorders, neuropsychiatric disorders, and pediatric disorders. Now, let us discuss the last part of our today's lecture, that is imprinting. Genomic imprinting is the epigenetic phenomenon by which certain genes are expressed in a parent of origin specific manner. If the allele inherited from the parent is imprinted, it is thereby silenced and only the allele from the mother is expressed. If the allele from the mother is imprinted, then only the allele from the father is expressed. Forms of genomic imprinting have been demonstrated in fungi, plants, and animals. As of 2014, there are about 150 imprinted genes known in the mouse and about half that in humans. Genomic imprinting is an inheritance process independent of the classical Mendelian inheritance. It is an epigenetic process that involves DNA methylation and histone methylation without altering the genetic sequence. These epigenetic markers are established, that is imprinted, in the germline, that is sperm or egg cells of the parents, and are maintained through mitotic cell divisions in the somatic cells of an organism. Appropriate imprinting of certain genes is important for normal development. The term imprinting was first used to describe events in the insect Pseudococcus nipae. In pseudococcids, that is mealybugs, both the male and female develop from a fertilized egg. In females, all chromosomes remain euchromatic and functional. In embryos destined to become males, one haploid set of chromosomes becomes heterochromatinized after the sixth cleavage division and remains so in the most tissues. Males are thus functionally haploid. Imprinting may cause problems in cloning, with clones having DNA that is not methylated in the correct positions. It is possible that this is due to a lack of time for reprogramming to be completely achieved. When a nucleus is added to an egg during somatic cell nuclear transfer, the egg starts dividing in minutes as compared to the days or months it takes for reprogramming during embryonic development. If time is the responsible factor, it may be possible to delay cell division in clones, giving time for proper reprogramming to occur. That's all about today's lecture. Hope you have understood it well. Thank you very much and goodbye.